thank you very much for these very kind words and also for your hospitality. I'm very, very impressed by what you are doing here. I understand, I just talked to the dean, that you are undergraduates and you organize such an international conference. I remember when I was an undergraduate, I didn't organize any international conferences. The command of English wasn't the same also, and also we didn't travel as much as you do. So I'm actually very, very impressed. So this new generation is completely different level. I would like to talk to you today about a cortical layer with no known function, but before I start, I would like to mention that Marta Krolak at the time spent some time in my laboratory, and I'm very happy to report that her paper, together with Dr. Fernando Garcia Moreno, was just accepted and it will be published very soon. So congratulations to her. And now Marta is Jankowska Krolak, and congratulations to getting married. And also I had visited Marta Wisniewska, also in my laboratory, and I was really happy to see how well her group is doing and doing extremely interesting work on thalamic development, molecular aspect. Now, I would like you to go back to the 17th century and walk down with me on this street here at Oxford. This is the main high street. This is where people get their degrees, the Sheldonian Theatre. And I want you to come with me to this road and have a look at this house. And this is the house where the word neurology was given to the world in 1664. So Thomas Willis, a well-known neurologist, published his book on cerebral anatomy, so the anatomy of the brain. And this was the first time when he used the word neurologia, neurology. So I consider Oxford the seat of neuroscience. Sorry, Warsaw. This is Oxford. And he is my hero, not just because of the word neurology, but because also he could find the proportions between research, clinical work, and teaching. He was an extremely prolific neuroscientist. He, with the exception of the last 9 years of his life, lived in Oxford. Then he moved to London because they offered him more money, and he practiced in London. But what he discovered was even more substantial. He was looking at brains of different species. Here I show you an example of a human and a sheep brain. And he noticed that the brainstem was very much the same. The proportions of the brainstem was roughly the same, and also the cranial nerves. He got, I think, about 10 out of the 12 right, which is quite a good ratio, even for a second-year medical student, usually on the exam. And he suggested that it's really the mammalian cerebral cortex, which must be the seat of the higher cognitive function, because you have a bigger cerebral cortex in human than in the sheep. And humans have the largest cerebral cortex. So he suggested that it's the cerebrum is the primary seat of the rational soul in man and of the sensitive soul in animals. And it is the source of movement and ideas. And he got it absolutely right in 1664. Indeed, it's the cerebral cortex which is responsible for episodic memory, language, initiation of movement. So he got this right. He had another line of argument suggesting why it's the cerebral cortex, which is the seat of higher cognitive function. And basically it was based on his neuropathological work. So he was looking at the brain of his patients, and he noticed that the brain of this boy had more but smaller sulci and gyri than this normal individual. But now, they didn't use fixative in those days, so both brains are falling apart. That's why it's open. But nevertheless, he was absolutely right. There are these polymicrogyria syndromes, which are brain developmental abnormalities, when you have more sulci and gyri than the normal individual. You can also have brain developmental abnormalities when you have smooth surface brain, and this is the lysencephaly syndromes. So probably in 1664, Thomas Willis was dissecting a patient with the polymicrogyria syndrome. And these patients have usually intractable epilepsy and learning disabilities. And his argument that the cortex, the cortical malformation was in the background was absolutely right. So now we know that there are lots of cortical abnormalities, and in fact many of them are so subtle that you don't even see a gross abnormality. If you look at the brain of a schizophrenia patient, or an autistic condition, or an attention deficit hyperactivity condition, you don't see major abnormalities in the brain. Nevertheless, these are all brain developmental abnormalities. And if you look at the numbers in the population, this is kind of staggering, just how frequent they are. And that's why I think it's very important that we understand how these developmental conditions and abnormalities come about, because the burden is quite severe. At the moment, I don't know what's happening in Poland, but in England, most of the money is going to neurodegenerative disorders, Parkinson's, Alzheimer's. But if you look at the disability-adjusted life year ranks of certain diseases, you see that preterm birth complications or neonatal encephalopathy was very high on this list in 1990, and these still remain very high in 2010. It means that probably not much progress was done in the understanding. So I would like to urge the funding agencies to consider these conditions and also consider the developing brain, because if you adjust these diseases, they have a bigger impact on society than many of the neurodegenerative diseases. And I understand these are horrible diseases, and we have to do something about those as well, but we shouldn't neglect these. So what I'm going to do today is divide my talk into two parts. The first part, I'm going to talk to you about the transient cell population in the developing brain, which is very important in sorting out the connectivity in the adult brain. And then in the second part of the talk, I will be talking about some of the remnants of these cells in the adult brain and argue that they have a unique function which we did not study so far. And that's why I gave the title, The Cortical Layer with Unknown Function. So we have to go back to the stage when the neural tube is induced and the neural plate will close. And if I ever get this to work, then all these fonts are very useful. But perhaps not. So we have to go back when really the earlier steps at neurogenesis, when you have a closed neural tube, and the rostral part of this will develop into telencephalic vesicle. And I shall be talking about mostly this part of the neural tube, the telencephalic vesicle, which will form the mammalian cerebral cortex. And you will see that this is the part which will grow the biggest. And there is a very close interaction between this unfolding genetic program, which is producing the brain, and also the environment. So, and this is the time when the brain is particularly vulnerable for all sorts of environmental interactions. And we take it for granted that we are born healthy and we have a functional nervous system, but this is not always the case. And lots of things can go wrong, and then subsequently this will influence the conditions. And some of these will manifest only in late teens, and that's why these brain developmental abnormalities have to be looked at. So I want you to come back with me to these very early stages, when you have a neural tube. This is the telencephalic vesicle, so this is a coronal section through a rat brain, but the human brain is very similar at these early stages. And then this is the side of the neuroepithelium, which is facing the lateral ventricle. So this is called the ventricular surface. And then the skull would be somewhere here, and the pia, and then this is the outside of the cerebral cortex. And you will see that most of the cells are dividing in the ventricular surface, and they will migrate out to form the first postmitotic cell layer, the so-called preplate, or primordial plexiform zone. And you can see the location of these divisions by using a phosphohistone H3 antibody, which is marking these epitopes which are only exposed in dividing cells. And you can see that most of the divisions occur at the ventricular and the so-called subventricular zone, and all the cells then have to migrate out towards the pial surface to form the cortical plate. So in all mammals, this is the common pattern. So the cells are born here in the ventricular, subventricular zone from these various progenitors, and then they migrate to form the preplate, which will split to marginal zone and subplate by the later arriving cortical plate cells, which themselves will form an inside first, outside last pattern. So the layer I would like to talk to you today is the subcortical plate or subplate. So these are the first generated cells in the mammalian brain, and they are very large, especially in primates, and lots of groups made enormous contribution to the understanding of this. The name itself is coming from Paško Rakić and Ivica Kostović. They called it subplate or subcortical plate. And Carla Schatz, Heiko Luhmann, Patrick Kennard, they made a huge contribution to understanding these cell groups, how they integrate into the brain. So without these cells, you could not form early connections. They are a large part of the cerebral cortex, and it's a very important part. So I became interested in the cell layer as a graduate student, and I noticed that these are the first projections which leave the cortex synchronously with the thalamic fibers. Then these pathways meet in the internal capsule and co-fasciculate with one another, and then the thalamic projections will contact these early corticofugal projections to cross through the paleo-subpaleo boundary. If these early projections are not in position, they cannot approach the cortex. They cannot get through. So they need these scaffolds. And subsequently, they will then follow these back to the cortex, and then the corticofugal projections from layer 5, for instance, they will go down to the cerebral peduncle, whereas the other projections from layer 6 mostly, and also from subplate, they go into the thalamus. So in all mammals, you have similar choreography to establish these connections, and the subplate, the earlier generated cell layer, is very important in these early steps. Now, after the thalamic projections reach to the cortex, it's not finished. The role of subplate is just beginning. These are the first projections into the cortex, and they target the subplate cells. So the subplate cells are the first transient target of the thalamic inputs. And if you stimulate the thalamic projections and you reveal the depolarization patterns in the cortex, you can see that the subplate cells are the first activated cell layers in the cerebral cortex. And you have this long-lasting, sustained depolarization pattern in the subplate. So I have been studying this using voltage-sensitive dyes. So I cut a section through the brain where you have the segment of the primary somatosensory cortex and the segment of the primary sensory nucleus here in the thalamus intact, and also the interlinking pathways. Then if you stimulate the thalamus, then you can reveal the depolarization patterns in the cortex, and you can see that the earliest depolarization is in the subplate layer. You have a little bit in the striatum also, and you can also see the spread of the action potentials coming out through the internal capsule reaching the cortex. These squares, they flash with 10 microseconds, so the time resolution is very good in this system. So these experiments revealed the spatial-temporal pattern of activation of the cortex, and now I want you to compare the postnatal 2 days old recording with a postnatal 4 days old recording. And you can see when you stimulate the thalamic projections, the 4 days old cortex will be activated much faster. It's a more columnar pattern. We are recording from the primary somatosensory cortex. And it is finished by the time the 2 days old cortex is activated. So you can see that within 2 days there is a huge spatiotemporal difference how the cortex is activated. There are all sorts of important changes. And if you change the sensory periphery in the primary somatosensory cortex by ablating the whiskers of those mice, then you will no longer have this plasticity. So all these spatiotemporal changes probably related to that. So what are these changes? There are lots of changes going on in these early stages in cortical development. First of all, the thalamic projections target the subplate and not the layer 4. It will take some time before the thalamic projections will reach layer 4 and they feed into these ultimate target cells, just like in the adult. Also, you have the transient subplate projections into layer 4, so they already stimulate the cortical plate before the thalamic fibers will reach them. You will have a transient circuit which will have subplate and also some somatostatin interneurons which feed into layer 4, but without this transient circuit, you cannot establish the ultimate circuit when these projections get removed and abolished. Subplate cells die off, and the somatostatin GABAergic interneurons, they remove their projections. And it was Andrea Marcus Smith, who is a graduate student in Simon Butt and my laboratory, who demonstrated these transient somatostatin projections. So all these changes are changing in the developing brain. And I want you to think about subplate cells as a dynamic scaffold which is essential for the building work. Once the building work is finished, then you can remove them. But they are absolutely essential for these early stages. Then you can reduce their numbers. So where are these cells coming from? Where are subplate cells generated? So most of the subplate cells are actually coming from the ventricular zone of the cerebral cortex, and they are generated from these radial progenitors. Others are generated and amplified by the subventricular zone intermediate progenitors. You have some GABAergic subplate cells arriving from the caudoganglionic eminence, and we discovered that some subplate cells come actually from the rostral-medial telencephalic wall, and these are glutamatergic subplate cells, but they migrate tangentially to the cerebral cortex. So you have heterogeneous cell population, and they have very different origins. And it was Fernando Garcia Moreno who developed technologies to study these early cell migrations and clones in the brain. And he developed methods how to deliver DNA into progenitors using electroporation. And this DNA was incorporated into these progenitors and was expressed in all their progeny. So when they were dividing, then they had labeled cells in the telencephalon. Now this is all very good, but then you don't know which one was coming from which progenitor. So if you want to know what is the clone size and what is the clonal dispersion, then you have to label them individually or have to be able to distinguish which one was coming from which progenitor. So Fernando decided to electroporate several different fluorophores, and then if you put different, like 15 different types, targeting the membrane, cytosol, or the nucleus, then you have a very unique combination of these dyes in these cells, and you have a fluorescent barcode. So just like when you go to the supermarket and you buy a pack of potatoes, you have a label on these potatoes, this barcode. So when you go to pay for it, then they just scan it in, and then they know that that's the potato and what's the price. So that's what Fernando wanted to do, to label these progenitors with a unique combination of these markers. And then all the progeny had that unique spectrum. And this is why he could then follow them in these clones. And he could distinguish between the different clones in the cerebral cortex. And from these experiments, we now know the clone size and also the clonal distribution. But what we really wanted to do is to do this just for the subplate cells. And we particularly wanted to know what's the proportion of the subplate cells which come from the subventricular zone. So not directly from the radioglia, but from the other layer of subventricular zone cells. Lots of groups claimed that only the upper layers were coming from these subventricular zone cells, but we suspected that subplate cells were also coming from here. So Navneet decided to have a look at this and took advantage of the fact that you have TBR2, a transcription factor which is selectively expressed in these subventricular zone progenitors, and he looked at the clonal analysis of these intermediate progenitors. So he wanted to know how many times they divide and whether they contribute to the subplate cells. And first he just crossed the TBR2 pre line with a reporter line, and then he counted the proportion of the labeled, non-labeled cells. And he was surprised to see that almost all cortical layers were going through from these intermediate progenitors. In fact, his estimate was for the different layers varying between over 40 to about 28%. And even in the subplate, you had a large proportion which was coming from these subventricular zone divisions. So indeed, subplate is coming from these intermediate progenitors as well as the radial progenitors. And that would explain also our previous finding in the TBR2 knockout, that there was a proportional reduction in all layers, including subplate, of the cell numbers. So that explains why we had these findings, and we now know that indeed all cortical layers are produced through these intermediate progenitors as well. And this is clinically important because you can have mutation in the TBR2 gene, and that's affecting your intermediate progenitors. And if you have TBR2 silencing in human, then you end up with the polymicrogyria syndrome. This is exactly the kind of brain I showed you at the very beginning from 1664 from Willis. So Willis probably was looking at a polymicrogyria syndrome where the patient had altered neurogenesis. And this information is also important when we want to understand other microcephaly syndromes like Zika. Where is Zika acting? Is it acting on the radioglia progenitors or the TBR2 positive intermediate progenitors or the outer radioglia progenitors? So suddenly all this became very interesting to study. And we and other groups are, of course, we are looking at these. So now just to summarize, so subplate cells, they come from the ventricular zone, directly from radial progenitors. They also come from the subventricular zone, and some of them come from this rostromedial telencephaly core. So how was this discovered that they come from here? If you put tracers into different parts of the embryonic telencephalon, then you notice that you can follow these cells as they migrate away. And Juan de Carlos in Madrid was doing these experiments, and he noticed that some subplate cells were labeled. So we teamed up and we had a look at this migratory stream, and indeed, a large proportion of the subplate neurons, they come from this part, and these are all glutamatergic, not GABAergic, glutamatergic subplate cells come through this tangential migration. And they express all these classic subplate markers. So the subplate is a very heterogeneous cell population. They have multiple origins, and it's a huge cell population in primate. Sometimes they're even bigger than the cortical plate, as you can see here, and you have subcompartmentalization. Like here you have an upper and a lower subplate, and you have a transient cell-dense band of subplate, which is primate specific. You don't see it in any other species. The mouse subplate is similar. It's a bit smaller. but nevertheless it's a good model system. But I want to emphasize that the human subplate is the largest one. There are unique characteristics here in subplate. If you look at the proteoglycan expression in human subplate, you see that this is a huge zone. Their water content is also different, and that's why you can image them in utero with MRI methods. And in fact, when clinicians talk about white matter damage, they really mean subplate damage. And this white matter damage is related to cerebral palsy. So that's why it's also important to understand the vulnerability, the selective vulnerability of this cell group. If they die off too early, you can't finish the building work. If you remove the scaffold before the building is finished, then you cannot complete this. And that's why it's clinically so important, the perinatal brain damage. The subplate cells, eventually they disappear from our brain, and it was demonstrated by Marla Luskin and Carla Schultz. They used birth dating studies in ferret, in fact. So they noticed that if you inject thymidine, when these cells are born, around embryonic 27 days, you label subplate cells. If you inject a few days later, you have layer 6 labeled. What is interesting is if you wait and look at these labeled cells a few months after birth, then they start to disappear from the white matter. Nevertheless, you still have a few left, and then layer 6 will stay in large numbers. So subplate cells are largely transient. They are present in large numbers in the developing brain, but then they drastically reduce their numbers. Nevertheless, you still have some cells, so-called interstitial white matter cells. So if you look at human brain, you will still see these cells. And in rodent, you even have a compact cell layer, layer 6B. So in human, if you follow these cells, actually Paško Rakic and Ivica Kostovic, they did it in macaque monkey. You can see that you still have labeled cells in the interstitial white matter cells, and these cells can have very important functions also in the adult. So now I want to switch to the possible adult functions. So you can see that these subplate cells are present in the 3-year-old human brain in the white matter, but you also have in the 40-year-old brain in these interstitial white matter cells, and you can label them with NADPH diaphorase. You can also use this marker in other species, but in human this is probably the best to label these cells. And it was Ted Jones and Akbarian who discovered that in schizophrenia you have larger number interstitial white matter cells and more widely distributed in the brain than in control individuals. And that really triggered my interest in the possible adult function of these cells in human. In rodent, you have these cells also, and they form a compact cell layer, layer 6B. So we started working on these cells about 15 years ago, and in those days you didn't have markers for these cells. It was very difficult to label them. We didn't know what they express, where they come from, and so we decided to use dissection methods to compare the dissected subplate with the cortical plate. In fact, I was on sabbatical, so I did some of these early dissections myself because it wasn't a very popular task to cut these out from postnatal brains. But later on, Wade C. Francisco and others, they developed all sorts of methods when you use laser beam to cut these out from sections. And of course, they did it much more accurately than I could do with manually. So from these studies, we now have very good including non-coding, long non-coding RNAs, and also isoforms. And we can use these now to address the function of the subplate cells. And Anna Horder, a graduate student at the time, discovered that the gene networks which are expressed in subplate, they contain a lot of schizophrenia susceptibility genes. In fact, more than any other layer in the mammalian cerebral cortex. So these gene networks indicated some clinical links. Later on, we repeated these studies in macaque brains together with the Allen Brain Institute, and my group was in charge of the subplate analysis. And this also showed very strong links with susceptibility genes for schizophrenia. So that's why we are very interested in the possible role of these cells, even in the adult. So you have a developmental cell population, and later on in the adult you have a much more reduced cell population, but that's also present there. So what is this adult cell population doing in the brain? Why do we leave some of these cells there? So that is what I would like to talk to you a little bit about. But before I switch to mouse models, I want to convince you that the mouse is a very good model because you have lots of shared genes expressed in mouse. Nevertheless, there you have human-specific genes which are not expressed in mouse, but you also have some mouse-specific genes which are not expressed in human. And even between mouse and rat, you have some differences in gene expression, so you should not consider the mouse as a smaller rat. There are two different species, and there are changes in the gene expression. And also, in Warsaw, you have the only colony of Monodelphis domestica, you know, this marsupial. Krystolievsky has this colony. And we also looked at the gene expression in the Monodelphis domestica, and they don't have a compact subplate layer. They have a more widespread subplate layer. Nevertheless, the markers are still expressed in this marsupial. So it looks like it's in all mammals you have these subplate cells. So some of the subplate cells survive and others will disappear. And we looked at what are the markers for the cells which stay on. And I just show you 4 here. And these markers reveal the subplate cell populations which will stay on after the first postnatal week in larger numbers than an average new and positive cells. So now we can use these markers to reveal the surviving subplate cells, and we can exploit this knowledge to look at the possible adult functions of these subplate projections. So what is the function of this layer 6B, this really thin layer in the brain in the adult? So when we look at the mammalian cerebral cortex, we are talking about this cell layer here. That's the layer 6B I'm going to talk about to you for the rest of the talk. So in the adult brain, this is a rat brain, you have a compact cell layer, and they are the remnants of the subplate cells. So if you burn the subplate, these will be left behind. And they are so different from layer 6A that Ramon Cajal suggested that this should be the 7th layer of the brain. And for a long time, we considered it as part of layer 6. Now, what might be the function of these cells? And I would like to argue that these cells probably involved in regulating what information is reaching the mammalian cerebral cortex. As you know, the sensory input is reaching the cortex through the thalamus. And in return, the thalamus is getting information from the cerebral cortex through the projections from layer 5 and layer 6. And I will show you that also from 6B. And in fact, if you look at the number of synapses in the thalamus, then these corticofugal projections outnumber the sensory input to 1 to 10. So you have more projections coming from the cortex. The brain likes to talk to itself. And Rodolfo Linach suggested that it's almost like the brain is always in a dream state, talking to itself, and ever so often you have a little bit of sensory input upgrading this image. So this is how he's thinking about it, and he might be right, that you have all this input which is regulating what kind of sensory input can reach our cortex, and how do we process this, and how do we focus our attention to certain stimuli but not for the other, and whether we keep some of our brain areas asleep focally than others. So I want to show you that layer 6B might be playing a role in this. And I was encouraged to look at the adult function of layer 6B by the late Ray Guillory, who had a huge contribution with Murray Sherman in understanding these corticofugal projections. So Ray Guillory and Murray Sherman suggested that you have two types of thalamic nuclei. You have the first order thalamic nuclei, they receive direct sensory input, and then they relay this input to the primary cortical area. Higher order thalamic nuclei, they never receive sensory input from the periphery. They get their input from the cortex and they relay this input back to other cortical areas. So they are involved in the cortical-cortical communication via the thalamus. So layer 5 and layer 6 has very different relationship to the first and higher order thalamic nuclei. Only layer 5 will communicate with the higher order thalamic nuclei. Layer 6 will communicate with both. Also, the kind of input they form on the thalamus is very different. Layer 5 projections, they are very powerful. They form large terminals on higher order thalamic nuclei. If you repeatedly stimulate them, you have depression. They mediate through inotropic glutamate receptors, and they are co-drivers. Whereas the layer 6A projections, they don't discriminate between first and higher order thalamic nuclei. They have much smaller boutons. They modulate the function of these cells via metabotropic glutamate receptors, and you have facilitation if you repeatedly stimulate them. So the two systems have very different properties. And the question I wanted to ask is, what is the role of layer 6B? Is it more similar to layer 5 or layer 6A? And now we can take advantage of these Cree-expressing lines where just a subset of layer 5 or layer 6 cells are labeled. And you can see, if you cross these lines to reporter lines, you can see the terminals of these projections. RBP4 is not expressed in the thalamus, so all these are coming from these layer 5 projection cells. And you can see that they leave the visual and the somatosensory primary nuclei free of label. So they don't target the first order thalamic nuclei, they go to the higher order thalamic nuclei, as Ray and Murray suggested. And you can also see that layer 6A will target both. So you don't have this selectivity. So we decided to look at the layer 6B projections, and we took advantage of all the knowledge we gained when we performed these transcriptomic studies of layer 6B. So we selected some pre-expressing lines which were selective for these cell groups, and we were surprised to see that they had selectivity. They did not grow into the LGN and the ventrobasal complex. So here I show you in more detail. They left the ventrobasal complex and the LGN intact, and they innervated the PO and also the LP, pulvinar. So they are very selective. They are not like layer 6A. To get a better understanding of the regional specificity of these projections, we used tree-dependent adeno-associated virus labeling from the different cortical areas, and we looked at the labeling pattern. And even in the generous injection to visual cortex labeled anterior association cortices. And we mapped out the cortical surface and the relationship to the thalamus. And to make a long story short, there is a selectivity. They don't like to go to first order thalamic nuclei. They always go to higher order thalamic nuclei in all cortical regions. And we used 3D reconstructions of these pathways. And also we were surprised to see that they have bilateral projections, and they also target anterior and lateral association cortices in addition to the thalamus. So these layer 6B projections are really weird. They go all over the brain, just like claustrum. They go to the thalamus, to the higher order thalamic nuclei, and also to the anterior and the lateral association cortices bilaterally. So layer 6B is a very interesting cell population, and it could have very interesting roles in the brain, even after many of them will disappear during the first postnatal week. And this observation I just showed you, that the visual layer 6B has a projection to the anterior cingulate cortex, was confirmed by the Allen Brain Institute when they did these injections to different regions of the brain, and they use beads. Then they sort the back-labeled cells, and then they noticed that if they inject to the anterior cingulate cortex, then you get 6B cells labeled, and they sequenced these individual sorted cells, and now we have markers for the cells which project there. And these single cell analysis will help us even more to understand the cell types which have these projections to the anterior cingulate cortex and also the higher order thalamic nuclei. But basically, we are now convinced that layer 6B is a hell of a cell group with these very extensive projections, which is very different from layer 5 or layer 6A. So we repeated many of these tracing experiments using the RBP4 and the NTSR1 Cree lines, and we now see these differences. But the question remains, are these drivers or modulators? Do they form large boutons, or do they form small boutons? And also, what is the relationship of these projections to the thalamic reticular nucleus? Regularly and Barry Sherman noticed that the first order thalamic nuclei, they always have a side branch to the thalamic reticular nucleus. So this is a sheet of GABAergic cells, and quite important cell group outside the thalamus. And also layer 6A will always have side branches to thalamic reticular nucleus, but not layer 5. So we wanted to know whether our layer 6B will have a side branch or not. And using the Cree-dependent adeno-associated viral tracing, we noticed that they usually don't have synapses next to these fibers crossing through the thalamic reticular nucleus. If you do the same experiment in PO, where they form synapses, you can see this opposition of the VGLUT1 immunoreactivity and these labeled cells. So we suggested that they don't make side branches there. And this was later confirmed in collaboration with Francisco Casca's group in Madrid, who can inject very small regions in the mouse brain using biotinylated dextrainamine. And if you look at the single fiber reconstruction, you see that there are no fibers in the thalamic reticular nucleus, and you have these branches in the higher order thalamic nuclei. And you can also see the difference. If you label a layer 6A projection, then they will have thalamic reticular nucleus side branches, and they target first order thalamic nuclei, so very different from the layer 6B. And if you label layer 5 projections, then they will not have side branches, and they have these huge terminals in the higher order thalamic nuclei, which is very different from the ones which are formed by the layer 6B. So all I'm saying is that layer 6A, B, and 5, they are different. They are heterogeneous populations, they all have different functions. And the layer 6B projections It looks like they have very small boutons, and they are always in this category. So to confirm this, we used electron microscopy, and we used post-embedding immunogold technique to label the TD tomato positive cells, which we used to label the cells. And you can see that the layer 6B projections are much smaller than the layer 5 projections. So they are very different. A layer 5 projection can have a huge specialized terminal with all these modifications, whereas the layer 6B is always small. So we discovered a new type of projection from the cortex to the thalamus. So what is this cell group doing? Is it driver or modulator? So for that, we have to label these projections with channelrhodopsin and stimulate them selectively and record from the thalamus. So these experiments are still ongoing, but it does look like that these are modulatory projections to the higher order thalamic nuclei. So this is a new type of projection with a very different pattern than the other cortical cells. Where are these cells coming? Where is the input to these cells coming from? So who is talking to layer 6B? When do they get active? To answer this question, we used monosynaptic rabies tracing. So we used the DRD1A Cree line so far to have a look at what was the monosynaptic input to them. So for this you have to inject a receptor, which is then getting expressed in a Cree-dependent manner, and then you inject a second time the rabies virus, which will only jump one synapse. So then you know what is the input to your layer 6B cells. So we did these experiments from the visual cortex, and we were really surprised to see how extensive the input is to these layer 6B cells in B1. They get input from the cortex, ipsi contra. They also get input from the lateral geniculate nucleus, and what we were really surprised to see that they also get input from the lateral hypothalamus. So there is a lateral hypothalamic direct input to these cells, and as you know, the lateral hypothalamus has lots of different cell groups, and we are now looking at which cell group this is exactly. And we suspect that some of these cells are actually generating orexin in the lateral hypothalamus, which have direct connections. As you know, orexin is a peptide which is important in arousal and the stability of arousal. If you don't have orexin, then you can have this condition of cataplexy, when you just suddenly fall asleep in the middle of a lecture. Or you can have these dogs, when they get excited, then they have this cataplexic behavior. So that's why we are so interested in the possibility that these lateral hypothalamic cells, they talk to a layer 6B cells. And it has been demonstrated previously that layer 6B cells have selective sensitivity to orexin. So Bayer and colleagues in Geneva demonstrated that if you record from different cortical layers and you expose these slices to orexin, then it's only the layer 6B cells which will respond to this orexin. The others don't. So layer 6B cells have a selectivity. And also, neurotensin, other peptide, has selectivity to these lower layers. It has been demonstrated by Christian Roberger in Karolinska Institute. So layer 6B cells can be in a very strategic position to mediate this lateral hypothalamic effect. More importantly, the higher order thalamic nuclei, they also show selectivity to orexin. So perhaps this system can be switched on and off in a state-dependent manner, and we are interested in the possibility that layer 6B is mediating this. Now I can't show you the results because we are still working on it. I just show you some preliminary data why we think that this might be the case. So there is quite an extensive literature on orexins and anxiety. There is a possibility that some of this is mediated through the projections through the locus ceruleus. But what we are interested in is whether this lateral hypothalamic effect is mediated through the layer 6B directly in the cortex. So for this, we decided to silence layer 6B by removing an essential part of the snare complex, SNAP25, and this way the layer 6B cells cannot release synaptic vesicles in a regulated fashion, so they are silenced. And we do that straight after postnatal life all the way to adulthood. And Luigi and Kim, they were looking at the behavior of these animals. So first of all, we had to confirm that indeed they don't have a regulated release. So if you don't have SNAP25, the synaptic vesicles cannot dock to the membrane and they cannot be released in a regulated fashion when the action potential is there. So to do that, we first of all we showed that the protein is missing. If you introduce Cree, we did that in vitro and also in vivo, so the protein is gone. If you use a Cree line, and also we demonstrated that if you remove SNAP25 in these conditional knockouts, then you don't have release. For this we use the synaptofluorine imaging method, and also we use Physiology since, but this method allows you to look at the release because the synaptofluorine will only glow when you have neutral pH at the time of release. You know that these vesicles are acidic, so normally the synaptofluorine will not glow. And that's what we could see in the knockout, that this glowing disappeared substantially. So there is no vesicular release, much reduced. You only have this spontaneous release in these animals. So we looked at various behavioral paradigms. There was not much behavioral change if you just look at the animals, how they walk in the cage. The locomotor activity was also very similar in the animals. Their sleep was not disturbed. And also, if you gave them a jet lag by advancing the timing with 5-6 hours, there was no change. So they had no problems with the sleep. The only differences we have seen in 3 separate anxiety tests that they had reduced anxiety. So if you have this elevated plasmase, they don't mind going out to the open arms in contrast with the normal controls, and they also don't mind going into the light box from the dark box and vice versa. So this suggests reduced anxiety. And they might have even reduced learning because if you put them into these Y mazes and you let them explore one arm and you block off the other, when you open it, the DRD1A Cree SNAP25 knockout will not go to the novel arm and explore this like the controls. It will be more of a 50-50. So they don't appreciate this novelty. So we are studying all these, but it will be very interesting to see how the activity of layer 6B is regulating some of the behavior. So what we are doing now is locally activating layer 6B or inactivating layer 6B using drags from certain cortical areas. So we are interested in the possibility that maybe just the frontal areas are more effective than the back for these studies. So what I wanted to show you today is that there are some cortical layers where we don't know the function of these cells. So the subplate cells are very important during development, but we shouldn't forget that you still have a thin cell layer, a remnant of subplate in the adult. And these are not just the developmental remnants. This is a proper layer with a proper function. And we have to understand these if we also want to understand some of these cognitive conditions where the increased number of these cells have been implicated. So we believe that this cell layer can have a very important function, how the brain is talking to itself. So this is where my laboratory is. This is where Le Gros-Clark was doing work on the thalamus. This is the Pitt Rivers Museum. Have you seen Harry Potter?
Yeah.
So some of it was filmed here, of course, and so we have the adjacent laboratory. We usually have coffee here in the biochemistry building. I'm very grateful to all the founders and all the people who were involved in the work over the decades. I'm very fortunate. It's incredible how gifted the new generation of students and postdocs are. We were nowhere then, 20, 30 years ago like they are today. They use lots of different techniques, and research became extremely interdisciplinary. So that's why I really like your idea to have this meeting as an interdisciplinary meeting. So anatomy, physiology, imaging, EM, behavioral studies, you have to do all these. In my time, we didn't do this. You could survive with one technique. But now you have to work in groups to be able to do this. And finally, I was taught this expression, the Polák Wenger Vabratanki Idu Shabli Idu Szklanki.
Thank you, Professor, for a great speech. And now discussion is open. So, are there questions? Yes? Maybe.
Thank you for a nice talk. My question relates to the experiment where you're looking at the time point at which the subplate cells disappear. So are you using staining of a marker to tell at which point they disappear?
So subplate cells are born between E10.5 and E12. So what you can do is you inject bromodeoxyuridine, which is a thymidine analog, and you give a pulse labeling at these stages. So we are using birth dating, and we combine it with various markers which we suspect that they label subplate cells which will survive in larger numbers. So, and also with some markers which we think that they disappear. So the data I showed you was demonstrating that if you birth date some subplate cells and you combine it with these markers, then they survive in larger number, those particular subplate cells with those markers, than the new and positive cells within subplate. So, but the peak of cell death in subplate in general is between P2 and P8. In human, you have a large number of substrate cells after birth, but within the first postnatal year, they largely disappear. In primate, there are very few studies, but Pashkoravic and Ivica Kostovic, now they reanalyzed their historic material from monkeys, and they have now some thymidine labeled data, quantitative data on subplate cell disappearance.
And also, do you know how heterogeneous this population of subplate cells are?
It is a horrible cell population to work with because they, as I showed you, they have various origins. So some of them, they come from the cortical telencephalic vesicle. Some of them, they come directly from radial glia. Others, they come from the TBR2 positive intermediate progenitors, 27% of them in the rodent. And then you have these tangentially migrating interneuron populations from the ganglionic eminence. And then you have the rostrotelencephalic wall. These are tangentially migrating glutamatergic cells. So it is really a mess, no? Because they are really, and what is really horrible, that after 15 years of working on these cells and the markers, I don't have a set of markers where I could distinguish these groups from each other. So you start labeling these cells with the markers which are really selective for subplate cells, like connective tissue growth factor, complexin 3, and then you start doing double, triple immunos, and there is always some kind of a partial overlap. So unfortunately, subplate cells are not like the interneurons. You know, when you will hear the last lecture from Yuri Bujaki from NYU, 20, 30 years ago, they were so lucky because they had parvalbumin, somatostatin, NPY, and then they could use these markers to distinguish interneuron. Subplate cells, unfortunately, it didn't work like this, but now, from single cell sequencing work, we know that there are roughly 5 different groups, but we can only define these with a combination of markers. So what the Allen Brain Institute did was, Bozilka Tosik was doing these studies, is that they dissected subplate cells, the subplate, and they sequenced 3,000 to 5,000 subplate cells, individual sequence. And then they looked how they cluster. And according to this, we have about 5 groups.
The reason why I'm asking this question is because once you start doing channel reduction experiments to look at the activity and try to assess the functionality of the cell population, it's very crucial to know, are you activating a heterogeneous population of cells, and thus, I don't know, making it very diffuse to try to link any causality between activity and
Exactly.
function.
So we will stick initially to the DRG1A3, which we know quite well and we characterize the projection. But that's just about 15 to 18% of the subplate cells. So that's a selective population. Your question is very, very relevant. Yeah.
Thank you very much.
Hi, I think I can probably speak loud enough. Thank you very much. Very nice talk. I was interested in this early phase when thalamus is only contacting subplate but not contacting layer 4. And I noticed that there wasn't any connection, at least drawn, to the PV cells. I was curious, I mean, in the adult brain, of course, the interneuron population is crucial for controlling excitability in layer 4 and other layers. I was just wondering, is there just not much activity coming in, or is there something else, for example, not much recurrent activity between the excitatory cells?
So that particular pathway, I'm not an expert in these interneuron and thalamic, but that's developing a bit later. The one connection I showed you, the layer 5-6 boundary somatostatin GABAergic cells, they are very early and they feed into the layer 4 and they receive direct thalamic input, but only the first couple of postnatal days. And around P5 they lose these connections and then the thalamic projections go directly to layer 4. So it was Andre Martha Smith in our lab, and also Gord Fischel at NYU had very similar studies, exactly the same findings, that you have a transient somatostatin, but not parvalbumin in those days. And then you get those projections slightly later. So there is an interesting choreography of establishing these. And of course, then you will have later on the parvalbumin cells as well.
Can I follow up with a short question? Because I remember when Patrick Kennard was giving a talk, but that was already 5 years ago, so he was mentioning that even in adolescence we still have the subplate in especially prefrontal cortex. And I was just wondering, is that, I mean, has there been a functional value added to this? Because at that time it seemed speculative whether that's, has to do with additional learning during that phase.
So this is exactly what we want to study, to what are these cells doing in the adult. And that's why I mentioned also that probably the front of the brain will be a bit more interesting than the back. So Patrick had a huge contribution understanding these early connections, but I don't think he did too much on the... He spent a sabbatical in my lab about 5 years ago. We dissected lots of early circuits, but nobody is looking at the adult subplate. And you are absolutely right, the front of the brain has more of these. So, and also more 4x positive projections go to the front of the brain. So we will probably start there. But nobody really quantified this in human. What is the proportion of the interstitial white matter cells in different cortical regions? According to Henry Kennedy, in the monkey, you have huge area differences in the interstitial white matter cell numbers. in different cortical areas.
Okay, maybe last question, because we are running out of time.
Thank you for the presentation. In what you show, it looked like neurons really knows where to go, axons. And they can go, I mean, from 6th level to thalamus and so on. I'm kind of new to biology of the brain, so I'm a physicist. And I really wonder, which is the biological mechanism, the trigger, or the path that the system of cues that leads an axon to its target into a brain, because it looks like a miracle.
It is a miracle, and I must say that I have two physicist sons. They didn't want to do biology. They both do theoretical physics, and in fact astrophysics, one of them. So, so if you look at the mouse brain, and you look at the projections from different cortical layers. So imagine you have a layer 5 cell which will have to go out, and it will have a side branch to the thalamus, the kind of big branches I was talking about. It will have also a side branch to the striatum. Then this axon will go to the spinal cord. It will have side branch to the basal pons, to the superior colliculus, and then finally it will find the target cell in the right segment of the spinal cord so that you can wiggle your finger. So isn't it a miracle to have all these motocopies and basically you sort out these projections. Now, if you look at the early development of these pathways and you look at the visual cortex and the same cell type, you know what's interesting? That they have exactly the same projections. They even go to the spinal cord from your visual cortex. And they also have projections to the superior colliculus, basal pons, and they have the thalamic and the striatal projections. So, but this is only present at the very beginning. And what's interesting is that this layer 5 projection will lose this to the superior colliculus from the motor cortex, and this layer 5 will lose the spinal cord projection at certain stage. So I would like to argue with you that they don't know what they are doing. So it's later on they will make sense of the environment, but initially they just follow some kind of a developmental algorithm, and then it will they have to make sense using cues. But how do they know whether they are in the right place? It's a very interesting question, because you have to have some kind of a differential gradient across the cortex of some molecules, that these cells differ from one another. They express different receptors because the cues are very much the same, so they have to respond differentially to these cues. So the topic you raised is a very interesting one. What are these gradients here to give areal identity to these layer 5 projection neurons so that they respond differentially to these cues in the brain? And you notice that I was using SNAP25 conditional knockout. So these cells cannot communicate to the environment. They cannot release synaptic vesicles in a regulated fashion. And do you know what we noticed? That they don't care. They still develop the projections correctly. So it's very interesting how this is all determined. So there's a complex interaction between these molecular gradients, cell type specific receptor expression, and these cues, and on the top of that you will also have this activity. So that's why so many things can go wrong. And you remember I started the talk that 1 in 100 of us will have schizophrenia, 1 in 10 dyslexia, God knows, 1 in 68 autism, and almost everybody has attention deficit hyperactivity according to this. So it is a miracle that we produce a functional nervous system which can meaningfully interact with the environment. But lots of things can go wrong. Thanks.
Okay, thank you one more time.